So I'm in a biostatistics department, and one of the latest things that we've ended up with, which a lot of other people also have, is microbiome data, human microbiome data. Is that better? So Jeannie Jackson, Katya Smirnova, and I have been trying to come up with ways to think about dimension reduction for the data, and then recently Di Cook gave us a couple of ideas, but all of the messes in here are mine. So what I'll do is give you an idea of what human microbiome data, what the structure looks like, what people use, mainly ordination methods for dimension reduction, and then this is sort of classical multivariate type stuff, where we know about Scree plots. Lots of people use Scree plots, but that doesn't really seem to work very well with these sparse high-dimensional data. And we really don't have any other ways that I know of thinking about this. So some of the other plots that we've been playing around with. We are hosts to huge microbial communities, and one of the latest things has been to try and think about the community composition, especially, sorry, I'm not that tall, especially in terms of how the community composition relates to health or disease, or even things, states such as obesity, et cetera. And you see, this has gotten a lot of press recently. So the question really is, what are the data, right? As data people, what we have, the data are really counts of DNA fragments. They're classified biologically. So depending on, just as the last speaker was saying, at which level these have been classified, and we've run into this problem with the data that we have, is some microbiome groups will actually have libraries that are, or data that are classified more finely at a different scale than with others. So comparing data across, from different labs and so on is also problematic. But basically these data are often called OTU tables. The term library size is used, this is really what we're talking about as a sample size. So basically we have a huge matrix. The rows are samples, meaning samples that we got from individuals, and the columns are what we would think of as variables are these various taxa. The challenge is there are huge numbers of taxa. So the gut microbiome, which has gotten a lot of press, has anywhere between 1,200 to 1,500 taxa. So you have that many columns. And usually we're dealing with human beings, and this is still relatively expensive. You're not getting that many samples. So there are data sets out there that have been analyzed where you had 20 samples and 1,200 taxa. So these counts in the OTU tables are often very sparse as well. So you have this library, this issue where, and it's a sampling issue, where some of the taxa give you huge counts and the others are very close to zero. So this difference in library sizes is a problem. So basically, even to do the first steps of just looking at the data, you need to do some sort of dimension reduction. And so what people have done is they usually end up with ordination methods for dimension reduction. The most common of these is principal components analysis, so either based on correlation or covariance. So some examples of where people have done this in published papers, and there's a huge literature now. The other thing that people will often do is take the counts, and because you have these very different library sizes and sparsity, et cetera, they'll transform the data matrix into presence-absence, so that you're just looking at zeros and ones. And then they'll use some sort of a distance matrix to then analyze that. If you have phylogenetic information, people will use phylogenetic distances. So these are the distances between the different taxa. So there are a couple of questions that we've sort of wrangled with. The first is, which transformation do you use? Do you keep the counts? Do you change them to proportions? There's a huge literature on people normalizing the data in different ways. And then the other thing is, once you choose your method, you could only really visualize in 2 to 3 dimensions. So people will often look at just 2 to 3 dimensions. So here's an example of a biplot. This came out of CHIME, which is one of the more popular pipelines to analyze microbiome data. And this is, as one of the earlier speakers put it, it's a hairball, right? This is like a hairball of balls. And we've had, even when you rotate, et cetera, it's really difficult to, especially when you're looking at taxa, it's really difficult to sort of get a sense of what's going on. But the bigger issue is we're just looking at 3 principal components, and these don't even explain 15% of the total variation in the data. So we're looking at a very small slice, and people are writing PNAS papers with this. So what we went back and looked at the ordination literature. And basically what you have is you have the original data matrix, and it's transformed in some way to get a matrix that we're calling H, which is then eigen decomposed. So the roots to H depend on the method that you're going to use, but then ultimately it's an eigen decomposition. And for PCA, for instance, the traditional tool is to look at a Scree plot. And you go back to the old multivariate books and they say, oh, 90% of the variation needs to be explained. Well, when you have these huge dimensions, 90% might be at 50, 60, 70. That's not going to work. And as you look at these Scree plots, especially for real data as well as simulated data, you don't have a nice break in the data or in the plot. So we started thinking of additional plots. So when you think of the eigen decomposition, all the Scree plot does is looks at the eigenvalues. Well, there's information in the other 2 matrices as well, the one that pre and post multiplies this matrix. And so we started looking at, what information is sitting in there. Why don't we look at it as we reduce the dimension, we're really approximating this matrix that we're eigen decomposing. So let's think of the errors and plot those. The other idea is this pre and post, these 2 matrices, they contain the coordinates of either the samples or the taxa in the geometry that you've now reduced dimension to. So let's look at how well those are being approximated. So basically, think of the whole thing, instead of just the eigenvalues and what their contributions are to the total variation, think in terms of approximating the matrix that we're decomposing and get to a dimension reduction where we look at the error in approximation as we change the dimension and sort of settle on something that's bigger than 2 or 3, but not as huge as whatever the rank of the matrix is. And then one of the later ideas was to use Andrews curves. So we had these intensity plots that got a little bit bulky to look at, but Andrews curves sort of project these into a nice Fourier, using a Fourier basis, and things look nice. Okay, so what I'm going to do is show you a bunch of these different plots. They're not glamorous in any way, but I think they get at different ways of looking at information. So before I move on to that, so one of the things people do is they'll take, so you've got your individuals and you've got their microbiome data, and what they will do is, in order to see whether disease states, for instance, relate to microbiome. So our obese are people that are more obese and less obese, are their microbiomes different? So what they'll do is they'll do this dimension reduction, and then they'll cluster individuals and then see whether that corresponds to whatever clinical covariate they're looking at. So taking individuals, reducing dimension, clustering the individuals to see if they fit into these covariate classes seems to be something, and it makes sense to be able to do that. So what we did is we take these various plots that we've started looking at, and then we cluster individuals. But we cluster them at 2 or 3 different dimensions that we pick using this information, and then pick the dimension where this clustering sort of becomes stable in a sense. Okay, so Scree plots, I think we know quite a bit about these. So this is simulated data I'm going to show you, where we have 58 OTUs. And first we simulated 100 tables where you had 25 samples and 58 OTUs. So 125 by 58 tables. The red plot is the theoretical Scree plot, the true one. And this is what the Scree plots look like. So there's a nice break here. This is the correlation matrix. So there's a nice break. The dimension of this, the reduced dimension should be 2, maybe 3, but the Scree plot comes down very gradually. This is the same story when you go to 60 by 58. So now the sample size is about the same as the OTU, as the taxa size. And things don't really match up until you have about 50,000 samples. There are no microbiome studies with 50,000 samples. If people get about 400 to 500 samples, and 58 is a very small taxa size. I mean, people usually have 400 and 500 taxa at a minimum. So we're using a technology that was derived for n much greater than p, and we're sort of just blindly applying this because we have n less than p. Same thing with the covariance matrix. You see similar stuff. Even at 20,000, things aren't quite matching up. So this I'll sort of zoom through this. So these are the different pieces of what I just described. Instead of just looking at the eigenvalues, if we start thinking in terms of approximating the matrix that we're decomposing, and here this index 1 through t is just the degree of the dimension reduction or increase. Same thing with the sample scores or the taxa scores. Let's try and approximate those using different dimensions and look at the errors. Just look at box plots. And we can also take these sample scores and project them and look at the Andrews curves and see if that becomes easier to look at. So these are all of the different data sets we've played around with, some real n greater than p, n less than p, et cetera. And we're seeing sort of the same sorts of things. So this Ravel data set is one that's been around for a while. This is what the Scree plot looks like. If we think in terms of approximating h, so on the horizontal axis is the dimension of the approximation, and on the vertical axis is just the errors of h minus h hat, for instance. And what you see is between the first and third quartile, everything's sitting right here, but you have a whole, you have some somewhere there's a lot of errors. So we haven't looked at all of the pieces of this yet, but this is sort of the idea that somewhere in here maybe is where we settle. Same thing with approximating the taxa and the sample scores. We can do the same sort of thing. And then for the, go on. Oh no, this is not. Okay, this might not, okay, so it's not, the PDF works in Acrobat. Oh, it does work, okay. It doesn't animate. Oh, it will animate, okay. So basically, we have Andrews curves at a higher dimension less than that, and then down here is the difference between the two. So if this works, what you're looking for is when this bottom one, it doesn't work. So basically, we cycle through these, and, sorry, we cycled through these, and okay, I'm going to skip this. So the variation that you see down there, by about 6 or 7 for the dimension, it basically just looks like sines and cosines. So the idea is where you don't see this extra piece, there is information there, so you could reduce down to that variation. So this animation has helped quite a bit. Okay, another thing that you see a lot is parallel plots. So people will have these parallel coordinates plots, and this is the sort of thing that it looks like, which is great. These are color-coded by a covariate that's useful for the vaginal microbiome data that Ravel had, except it becomes really difficult to tease anything out of this. So what we did is, here is a dimension reduction of 3, here's 6, here's 11, and all we're doing is clustering individuals at those fixed dimension reductions. So the idea is, what's the stability? Does this group, is that the same as this group? Is that the same as this group? So somewhere there will be, we think, stability, and that's the dimension that maybe we work with. This is the same information, but now we're just looking at cluster membership. And we sort of settle at 6 in this case, because there isn't that much of a difference between when we go from 6 to 11. And we can also do an intensity plot and look at this cluster membership to see if the samples, if they've changed dramatically or not. So, okay, a different use of intensity plots, and this has been helpful when we just look at a handful of taxa. So here on the horizontal axis are certain taxa that are of interest in this vaginal microbiome data set. So there are lactobacilli that people are interested in. And so what you have is on the horizontal axis are the number of principal components. So when people look at things in 2 and 3 dimensions, all they're doing is looking right here. So you have some taxa that the darker color means they're farther away. The lighter color is closer to the origin. So you have some taxa that are very dark at the beginning in 2 and 3 dimensions. But then after a while, they just fade away. And so if you reduce dimension too much, maybe you're sort of throwing these taxa out as not being big players. So I think this is something that needs to be looked at a bit more carefully. And then there are others that start out very light and then show up and they're very dark. So they become bigger players as you go farther out. Okay, so basically this is sort of our recap, and then I just have one more slide. Scree plots are all we've been using because that's what we've known. But maybe looking at the amount of variation captured is not the only criteria that we should be thinking about. We can think of this in terms of approximating the matrix that we're decomposing. We can think of this in terms of approximating the information in the sample and taxa data as we reduce dimension. And then looking at different types of things like the Andrews curves that can help you sort of tease out what might be there. So I'm going to zoom through. We did this with the other methods that people use as well, which was PCOA. But here's sort of the punchline, I think. So we took one data set, and we've done this with all of our data sets now, the simulated as well as the real. And there were two questions. One was if we use two different methods, if we use principal components where we keep the original counts and change them to proportions in our case, and we take the same data set and make it presence-absence, do we end up with the same sorts of results? Do the individuals that cluster by looking at presence-absence of taxa, do they also cluster together when we look at proportions? And the part that's disconcerting is 6 out of the 32 samples cluster the same way. And this is using different dimensions. So for PCA, we settled on 10 dimensions. For PCOA, we settled on 6 dimensions. On the other hand, the plots that you see in publications at most 3 dimensions. So we just take 3 dimensions and we compare PCA versus PCOA, and 8 out of 32, the cluster memberships agree. So when people are taking their samples, clustering them using some drastic dimension reduction, and then saying, these covariate classes match up with these samples, and here we have a publication that is worth looking at. I think we need to come up with sort of better ways of thinking about this dimension reduction. So this is more or less what I have. I don't have any solutions yet. I think I've become very cautious when I hear these various studies. I mean, one of the ones that really bothered us in the Ravel data, and this is US data, they have 4 ethnic groups. And over, it's the vaginal microbiome of black women is different than that of other women. And it's one of these things where you go about more than 3 dimensions out, everyone looks the same. There really isn't a difference. And so, this kind of stuff makes me nervous. So anyway, this is basically all I have to say. I think But I think if anyone has some nice ideas for how to visualize things in higher dimensions, this would be great. Yeah.
Thanks, it's a really interesting talk, and it's great to see this kind of methodological development, which I think is a little bit unsung and underappreciated a lot of times in public microbiology. So one thing I've heard, I don't know a lot about random forests, but I heard that they are good at this small n high p problem. I kind of think about how you might apply them to this kind of data set. Like, would you just drop out OTUs? Would that be how you look at different things? But
No, a lot of times when people use random forest, see, one of the other solutions to dimension reduction is people will go the biology route and come up with diversity indices. And then they'll use those, and that'll be one of the covariates. But yeah, I don't see a different piece, and this actually has to do with networks, is I have colleagues who are biologists who'll say, and it makes sense, these bacteria or taxa don't live by themselves, right? They're in little groups. And so we really need, but it's really hard to think of how to build networks when you don't have that many samples and you have that many taxa. Sparse as well, yeah. So there are loads of, the bigger problem that I didn't even touch on in this, there's a lot of data filtering that happens even before you get the data. There's a huge amount of information that's thrown out because we don't know what taxa they are. So unless you have somebody that's interested in doing de novo research, they're not going to sit in the lab and try and figure out what this is. So that's the first one. The other thing is because there are so many errors in the sequencing and so on, counts that are really close to 0 will often get filtered out. And, I keep thinking, sometimes it's the small but dangerous stuff that gets you, right? So are we throwing out some of these things because they're close to 0 and we don't know what the errors are? So there are contamination problems. I mean, it's just, it's fast, it's like full of problems to solve, but it's sort of scary. Yeah.
Just wondering, in the machine learning literature, there's an algorithm called T-SNE, which is quite popular. I think
Say it again?
it's called T-SNE.
Yes, that's right.
I'm just curious, I think that specifically targets sort of like a 2D or 3D representation that
Right.
has some clever thing which minimizes it, but I've heard it has some problems as well, but I was just wondering if you come across that.
Yeah, we haven't, Doug Cook has suggested this. We haven't gotten that far as far as trying this yet. I mean, I think what I'd like to do is to try all of these different methods that people have and sort of compare them. And that's on the list. I've seen more and more posters on that at conferences more recently. Yep.
Same thing for non-negative matrix factorization.
Yep. That's,
I don't think it's going to help, but
it's not, no, actually Katya Smirnova, so she tried that. Early on when we started with this, she was, non-negative matrix factorization is going to do the trick.